Next talk comes from Cenya Imaging, will be given by James Simon, and it's entitled Clinically Applicable Nanoparticle Platforms for Cell Tracking and Other In Vivo Imaging Applications.
Okay. Right, I'm going to talk to you today about Cenya Imaging. Cenya is a company that's in the process of being spun out of Radboud University Medical Center in the Netherlands. And the work I'm going to talk about is that of Mangala Srinivas and her group at Radboud, where we're developing contrast agents to track cell therapies in vivo. So this is a slide which shows you what's going on at the moment in the cell therapy field, so particularly the adoptive cellular therapy field. In the center there, the bullseye, the two products that have been approved for marketing. And as we go outwards in concentric circles on this target, you see more and more products that are in development. So the earlier stages are further out there. So what's going on is that there's a wave of cell therapies that's entering the clinic and is going through the clinical pipeline. So this just captures adoptive cellular therapies. There's also huge volumes going on in regenerative medicine, and dendritic cell vaccines are also making a resurgence. So the problem that we're looking to tackle is that cell tracking is needed for cell therapy development. All of these different cell therapies have one thing in common, and that is that they're harvested from donors, they're activated in some way to make therapeutic cells, and then they're put back into people. They then move to their site of action and do their work. And generally speaking, the more cells go to the right side of action, the longer they stay there, the better the outcome. The issue is that in humans, we have no way of determining this. It's very difficult to do that over any period of time. There are some examples where it's been done, but it's quite difficult. There's a lot of different options preclinically, but they're not translatable for safety reasons. And when we've spoken to a number of people about the efficacy reasons for doing this and the safety reasons, we've been told things such as ability to quantify cells over extended periods of time in humans is the holy grail. And it's likely that regulators will mandate biodistribution studies in humans once the technology is available to be able to do this relatively easily. So from both efficacy and safety perspectives, there's an unmet need for a technology to quantify cells over extended periods of time in humans. So our solution for this are Cenya nanoparticles to label and track cells. These are quantifiable, they're stable, they're translatable to use in humans, and they're multimodal. So the basic nanoparticle was designed initially for use with 19F-MRI. It has a PLGA shell and filled with a liquid perfluorocarbon. The liquid perfluorocarbon works as a contrast agent for 19F-MRI. So fortuitously, it also works with ultrasound and photoacoustic imaging. So it gives very good contrast with these other types of imaging modalities. And furthermore, we can easily adapt these by adding radioligands for SPECT and PET, by adding gold for CT, and for fluorescence imaging with dyes. And fluorescent dyes also enhance the photoacoustic imaging and make it far more sensitive. Importantly, all of the components that are in these nanoparticles are approved for use in humans in far higher doses than we currently use them. And we've had now 2 nanoparticles that are approved for use in a dendritic cell clinical trial. That's going to happen later this year. These 2 nanoparticles differ in that they have different perfluorocarbons in them, which means that we can differentiate them using 19F-MRI in vivo. And they also contain different fluorescent dyes, which means that subsequent histochemistry allows us to also differentiate the nanoparticles or whatever cells they've labeled. So the clinical trial is going to label 2 populations of dendritic cells. We'll use ultrasound to make sure that they're injected in the right location, because for dendritic cells, if they're not located in precisely the right location, they don't work. We will then use 19F-MRI to track and quantify the cells, and then excise lymph nodes and use histochemistry to validate the data that's seen from imaging. So this technology, so the nanoparticles label a wide range of cells without altering their function. So here's just a subset of the cells that have been tested. So dendritic cells and natural killer cells, mesenchymal stem cells, also activated T cells, which I've not included here. So importantly, they do not alter the function, and a variety of tests have been done to do that. And we had to make those tests to get the clinical approval, or the approval for use in the clinical trial. They enable in vivo quantitation of cells. So I noticed this was shown earlier on this particular image, so some of Mangala's work. So in this panel here, you see the 19 fMRI of T cells that have been put that have been labeled and put into mice, and this is superimposed on the regular MRI here, so you can see where the cells have gone. We're able to quantify those cells, and the reason for this is that 19 fMRI, as was eloquently explained earlier on, is linear, so it allows that quantification, and that tends not to be the case for some of the other more sensitive techniques, the iron oxides. So cells can be quantified in vivo for a period of weeks, and this is something that's actually, which we think is unique about this. So this is an example of a mouse that's been injected with labeled activated T cells, same mouse, imaged over 3 weeks, where we can continue to quantify those cells. As long as those cells are alive and haven't released the perfluorocarbon, we can image them depending on how far they have divided and how far the label has diluted. This is not the same animal or the same experiments, a variety of experiments, but it's the same nanoparticle, which shows the multimodality here of one single type of nanoparticle. We can use fluorescence imaging, and this is just the lymph node that's been excised, but this is in a mouse leg. 19 fMRI, this is the same leg. Ultrasound also allows you can use ultrasound with these, and then optoacoustic imaging, and in green there you can see the label in optoacoustic imaging. The PLJ nanoparticles can also be readily radio-labeled, and we've done this with indium and can also do it with zirconium. And this means that you can use SPECT to track cells after they've been injected into people and into animals in this particular case. So you can see quite a lot of the label is around the mouth, and that's because the mice, after 24 hours, and that's because the mice lick the area where they've been injected. And you can see all the various lymph nodes that are labeled here. After 1 week, we can still see a little bit of label, but it's degrading quite fast, and after 2 weeks, it's a lot harder to see. So the clearance of the nanoparticles is relatively rapid. So this is an experiment in which mice were injected with nanoemulsions, nano capsules and nanoparticles. And then we track to see how the perfluorocarbon is cleared from the liver and the spleen. And so what you can see is that the half-life for the nanoparticles is a lot faster than for the other types of delivery methods. And that's important from a safety perspective. The reason we think that this is happening is that this is a very unusual structure. The agent has a unique fractal structure, and what that means is that it degrades into smaller particles when it's released from cells, and that allows a fairly rapid clearance both locally and systemically. So this again helps us in persuading regulatory agencies to allow us to use this in humans. And no toxicity has been detected in vivo. So this is huge doses that have been given to mice, up to 60 milligrams, and at the top is the liver and the bottom is the spleen. So in summary, there's an unmet need for a technology to track cell therapies in humans. Solution that we're developing are Cenya nanoparticles. They're quantifiable over time. They're translatable to humans. They're safe. They're manufacturable. We can manufacture them to GMP grade at low quantities at the moment, but we also have continuous flow at non-GMP, which we can transfer to GMP in due course. They're stable as a powder for at least a few months. That's the only formal studies that we've done. We actually use these for years. And also they're multimodal. So one of the aspects that I didn't mention earlier on is the multimodality allows, if we talk to people that are developing cell therapies, they all have different questions that they need to answer in their studies, and this allows us to use the right imaging techniques that will address their particular questions. So the market dynamics are favorable. We're seeing very rapid growth in cell therapy development. So 20 to 40% growth per year over the last 5 years in the numbers of clinical trials that have been started. And there's limited competition in this area. So we think there's an interesting area to commercialize. We have good patent protection in the EU and Japan. And one of the aspects that I've not really touched on here is this is really a platform technology. So we're using this in the first instance to label and track cells, and that's because the regulatory perspective there is the regulatory pathway we've defined is fairly easy, fairly low hurdles, but there are other applications of this. As I said, it's a very good agent, contrast agent for photoacoustic imaging and also ultrasound. And we think that it may well play a part in contrast enhanced ultrasound in the future, and potentially in photoacoustic imaging as photoacoustic imaging moves towards the clinic. So acknowledgements. So this is the work, as I said, of Mangala Srinivas at Radboud, funding from the ERC and NWO in the Netherlands, and also at the moment these particles are being tested by the EU NCL as well, so our thanks to them. Thank you.
For this interesting presentation, questions? Okay, I have one. The multicolor fluorine MRI, do you have any, so basically the two different particles, do you have any clinical applications in mind? Because for just following the
Yeah, so that was used because in the clinical trial that's going on, they're testing two different dendritic cell populations. And they wanted to see where those dendritic cell populations go and whether they migrate differently. So you can imagine potentially in early stage clinical trials or non-clinically as well that you may want to compete different cell types, or you've tweaked them in different ways to understand which might be your lead candidate. So there's a potential there. So anything where there are multiple cells that are being put in, so yeah, you have possibilities.
And the fluorine compounds, are they single peak fluorine compounds?
So one is a single peak, one is a multiple peak, but they don't overlap. Yeah, so I'm a commercial guy.
Okay, yeah.
But yes.
More questions? No? Then thank you very much for your presentation. And I would... And I would also like to thank all the other presenters and the people who have actively participated in the discussion, and I would like to close this session. Thanks.
Doesn't want me to speak anymore, yeah. Well, thank you for the brave of you still there. If you can stay for 3 more minutes, the jury will just take the final decision and we will have the closing ceremony with the awards for best talk and best book. So please stay for 3 minutes and we'll be back. And the party, no, and it's over. Thank you, sir.